Welcome back, guys. I think my connection is okay. So yesterday we completed cells, and I told you guys that I'll be starting the cell membranes chapter next, but I think we should first do the past papers and question answer sessions, because if anything, any point is left in this chapter, if any query is left for me to address, this could be addressed in this part of the solving of question answers, yeah, or you can say MCQs. So let's just solve together, because it would be a practice for me too and for you guys too. So let's just start doing the question answers. So here we go. So yeah, what is okay? So let's go. I think my screen is visible, so we're gonna be doing biological molecules first. And the first question is, what is general formula of amylose? Here, am I audible? Yeah. So what is the general formula of amylose? So as we have discussed, you need to recall the types of carbohydrates. So there were three types, yeah, main types: monosaccharide, disaccharide, and polysaccharide. Yeah. In monosaccharide, we talked about glucose, fructose, galactose, these three. Then we talked about, in disaccharides, about maltose, sucrose, and we talked about lactose. Yeah. So they were basically the disaccharides. Maltose was formed due to glycosidic bonds between two glucose, whereas fructose was the formation of glycosidic bonds between fructose and glucose, and lactose was formed from the glycosidic bonds between glucose and galactose. So the next thing that we discussed was the hold on, let me just do not disturb mode here. So the next thing that we discussed was the polysaccharides, in which we talked about cell wall structure, which is cell wall components, which is cellulose, which is a kind of polysaccharide. We talked about starch, we talked about glycogen, we talked about N-acetylglucosamine, which is another part of peptidoglycan, which is found in prokaryotic cell wall. Then we further discussed the prokaryotic cell wall in the chapter of cells. Anyways, in the polysaccharide, where we discussed about starch, we also discussed that starch is made up of two main components. One is amylose and another one is amylopectin. And amylose was a straight chain polymer. It was not branched, and the thing is, it was the polymer of glucose, whereas cellulose was polymer of beta glucose. This is the polymer of alpha glucose. Yeah. Now, this question seems very easy, but there is one technical thing that students do wrong while attempting this question. So first we see here that what is the general formula of amylose. So as we know that C6H12O6 is the formula of the glucose, and because it is made up of glucose, the student see, oh yeah, this is C6H12O6, this is the formula of glucose, and they, in a hurry or whatever, they just mark this one out. But this is where things get interesting. This is not the answer here, because Amylose is the polymer of glucose, and when polymerization occurs, H2O or water molecules eliminates. Yeah, so because of the elimination of water molecules, C6H12O6 is not C6H12O6 after the elimination, because water is eliminated. So let's just say this is a typical glucose molecule. Yeah, now the formation of bonds are at this first and that last, OH 1 and 4th. So the formation of glycosidic bonds is between the 1 and 4th. So let's say, for example, from the side of 1, OH is eliminated, and from the side of 4, H is eliminated, forming H2O. So because 2H and 1O was eliminated for the formation of glycosidic bonds, the H is gonna be 10 and O is gonna be 5. So the final answer is gonna be this, D. Next, which statement describes how the molecular structure of starch is suited to its function? So it is talking about starch again. Starch, recall, is made up of amylose and amylopectin. Amylopectin is not as heavily branched as glycogen, but it is branched structure, 1,4,1,6 branches, 1,4,1,6 bonds, glycosidic bonds, and amylose is straight chain polymer, which is 1,4 glycosidic bonds. So amylose has branched structure. This statement is wrong. So A is wrong here. In the breakdown of amylose and amylopectin, many condensation reaction release stored energy. Now, recall, condensation reaction was not involving the breaking of bonds, but it was otherwise. Condensation reaction was the formation of bonds. It was hydrolysis which accounts for the breaking of bonds. So this one is wrong too here. Another one is, in the formation of amylose and amylopectin, many hydrolysis reaction allows the release of stored energy. This seems to make sense here. Anyways, we're gonna read the D1 too. Amylose and amylopectin form complex that is insoluble and does not affect the water potential of the cell. Now, one thing you need to realize here is that C is also wrong here, because in the formation of amylose and amylopectin, many hydrolysis reaction occurs. Formation would never require the breaking of bonds. So formation of amylose and amylopectin is due to condensation reactions here. So formation of amylose and amylopectin does not involve hydrolysis reaction, so this is wrong too. And the B part was wrong, because in the breaking down of amylose and amylopectin, many condensation reaction occur. So breaking down of amylose and amylopectin was the hydrolysis reaction, and the formation of amylose and amylopectin was the condensation reaction. So these three are wrong. So we're left with D. And this is the elimination method that I used for solving the MCQs, but you can, but it was the last answer. If it was A, then we would have considered it and moved on to the next ones, because a lot of time in CIEs, they don't usually give the wrong answer. They sometimes give the best possible answer, and you have to choose that one. So for example, there's a question which includes 4 options. 1 or 2 are going to be wrong, but the rest of the 2 are going to be correct. But the thing is, according to the situation given by the question, you need to choose the one that suits best. So these are BCQs actually, so you need to read carefully all of the options. So moving on to the next question, which is cows and whales are mammals, makes sense, that produces milk to feed their babies. Newborn whales grow faster than newborn cows. Okay. Milk contains fatty acids with a range of chain lengths. The table shows the percentage of fatty acids of different chain length in cow milk and whale milk. So the chain of length of fatty acids, number of carbon atoms. So in the cows, the percentage of 4 to 12 carbon containing fatty acids is 22% in cows and 0% in whales, whereas the highest percentage within whales is greater than 18 number of carbon atoms in a specific fatty acid. So okay, let's just read the question. Which statement about the ratio of short fatty acid 4 to 16 carbon atoms to long fatty acids 18 or more carbon atoms in the milk of cow and whale is correct? The ratio in cow milk is higher because newborn cow needs more energy than newborn whale. Who knows that? I mean, how can you say that? The ratio in cow milk is lower because newborn cow needs less energy than newborn whales. I mean, let's just see that. The ratio in whale milk is higher because newborn whales needs less energy than newborn cows. The ratio in milk is lower than because newborn whales needs more energy than newborn cows. So this makes more sense here because the ratio of carbon 14 to carbon 12 is more in the newborn, is less in the newborn whales because they need more energy than the newborn cows. So they have a higher percentage of the fatty acids in their milk. So next question is, the structure of phospholipid includes polar phosphate head, hydrophobic fatty acid chains, saturated fatty acid chains. Now this is a very easy question. You need to do it yourself. I'm giving you 2 seconds to think the answer. So the phospholipid structure includes polar phosphate head. So number 1 is correct, which means C and D are wrong, because the head is hydrophilic, and anything which is hydrophilic, most of the time it is polar in nature, because polar and polar attracts each other, and water is polar itself. So next is either it is 1 and 2 or it is 1 and 3. So the structure of phospholipid contains hydrophobic fatty acid chains. Now this is correct. And the next one is saturated fatty acid chains. This is wrong because one of the chain in the phospholipids is unsaturated too. It means it contains double bonds. Yeah. So 1 and 2 is the best possible answer here. So we're gonna go with that. Next question is, the diagram shows the molecular structure of peptide. Which molecule would result from the complete hydrolysis of polypeptide? So this is the structure of polypeptide. Now remember, what was the structure of polypeptide? It was NH2 CRH, and then there was carboxyl group here. So this was the structure of the amino acid. And if we remove OH and H from this side, the remaining structure was the part of the polypeptide. So we need to see here that which accounts for the structure of polypeptide here. So NH2 CH, this is R group, and then it is C double bond O and this. So this is one of the amino acid. This one is another, and after that this one is another. So these are different types of amino acids here. CH3 C double bond OH. So this one is correct for this representation. Here we had NH2 CH, CH2 and OH and C double bond OH. So this one accounts for this. And NH2 CH, and after that CH2 C double bond O, CH2 CH2, no. So this one is wrong here because there are two CH2s here, and whereas there was one CH2 here. So this one is wrong. We need to see that NH2, this is NH2CH, NH2CH, CH2 here, C double bond O, NH2. So this one's correct here. And again NH2CH, CH2, C double bond O, and NH2. So this one is correct too here. And if we talk about this one, so NH2CH, CH2, C double bond OH, and C double bond O, NH2. So these three structures are correct here. So let's just mark it. So this one is wrong, this one is right, this one is right, and this one is right. Now we need to figure out the rest from the rest of these three, which of the two are correct. So let's say NH2 CH, CH2 and OH, C double bond OOH. So the next structure that we have here is wrong. So moving on to the next one, NH2 CH, CH2 OH. So this one is correct. NH2 CH, CH3 and C double bond OH. So in this case, C is the answer. Because here it is only C, H, and the D is also wrong. Moving on to the next question. The diagram shows hemoglobin molecule, beta globin chains, alpha globin. So as I've explained in the videos, that a hemoglobin molecule is made up of 4 chains, 2 alpha and 2 beta, connected, and it also contains iron. I think this contains 512 amino acids as far as I remember, containing 123 and 128 in alpha and beta chains respectively. I'm talking about the amino acid number here. So which row identifies the part of molecule of this molecule? So let's see here. This is part 1, number 1. This is number 2, and this is number 3, and this is number 4 here. And this is number 1. So number 2 here shows us kinks or turnarounds. So it's alpha helix structure, yeah. Whereas if we talk about the part here, it's beta plated, and the part 1 is the heme group. The thing is, the picture is not very clear here. So I'll explain this question in a bit because I need to extract the original image here. It is very bad right now, but we have alpha helix here. So this part is alpha helix because it has this kinks, or you can say round structure, whereas the fourth one here is the hydrophobic group because it's inside. As you can see, it's folded inwards because it is hydrophobic. If it was hydrophilic, it would have been outside. So number 2 is hydrophilic, number 4 is hydrophobic, and the B is oxygen binding site. So the best possible answer here is B. Which color indicates the lowest concentration of reducing sugar present in a solution after testing with Benedict solution? So this is the part of the practical analysis, or you can say paper 3, which is alternate to which is practical, but paper 5 is alternate to practical. So this is a part which is required in the practical skills. I haven't done that yet. As I've told you guys, I will do it in the next class before starting the membrane. I will just go briefly over the practical skills from the chapter of biological molecules and from the cells. In the cells, you were required to know about the image magnification and stuff, how to calculate the magnification or actual image size, etc. Those were easy things. And in the biological molecules part, you need to know what are the colors. For example, let's say starch test. You need to know some tests here. One is starch test, where iodine is used for the identification and the color is black, blue-black, you can say. Another one was lipid emulsification. Another test was Benedict solution, which is the testing of glucose inside the solution, or you can say reducing sugar test. And the final result of that is brick red. And there is series of color changes, but the final color, if the concentration of reducing sugar is high, it is brick red. And another test is biuret test, which is protein test. So we will learn about this, so there's no need to attempt this question right now. The diagram shows the monosaccharide xylose. Okay, this is a concept-based question here. Many xylose monomers can be joined by glycosidic bonds to form polysaccharide, which is found in plant cell walls. Okay. Which diagram shows the formation of glycosidic bonds between two xylose monomers? So now here what is done is that they have introduced a new thing which is not in the syllabus, but the concept required for you to understand, or the concept that you have built on the formation of glycosidic bonds, is similar here. So let's say this is number 1 carbon and this is number 4th carbon, and they OH from number 1 and the OH from number 4th both reacts with each other, forming the glycosidic bonds. So what's going to happen here is another molecule which is going to mimic, let me just make this out here. So for example, this is a molecule here that we have. What am I doing? So this is a molecule. We have made a duplicate exact replica. What's going to happen here is this is going to get inverted. This is going to get inverted. You know why? Because the OH bonds needs to be in front of each other for bonding, yeah. So in order for the formation of 1,4-glycosidic bonds, this image is inverted, and we need to find this image. So there is OH, and so as you can see, D1 resembles the image that I've made upside, yeah. So question number 9, which molecules are monomers? So ribose, glucose, deoxyribose, and sucrose. So ribose is any sugar which is 5 carbon, yeah. It is basically in DNA, 5 carbon sugar. The type of 5 carbon sugar is, for example, ribose. So it's a class. It's not a monomer. Ribose sugar is not a monomer in itself. If you talk about, oh, hold on, what am I saying? Ribose is 5 carbon sugar, like it is in deoxyribose. I'm sorry for misunderstanding here. Anyways, deoxyribose or ribose sugar in ribonucleic acid. So this one is a monomer. Glucose is a monomer, as you know. Deoxyribose is a monomer too, and sucrose, on the other hand, is not a monomer. It is the it is the disaccharide of glucose and fructose. So if you cancel out the 4, so we're left with 1, 2, and 3. Which molecule have properties that are dependent on hydrogen bonds? So cellulose has properties that are dependent on hydrogen bonds. Water molecules are highly dependent on the hydrogen bonds. So we are definitely sure that 1 and 4 are the answers here. So 1, 2, and 3, it does not contain 4. 1 and 4 are here. 2, 3, 4 are not here, and 1 and 4 are here. So we need to choose between those two. So hemoglobin. Does hemoglobin involves hydrogen bonds in its structure? And the answer is yes, it does. It's a protein structure. It's a you can say tertiary structure, and in tertiary structure there are numerous hydrogen bonds. We will confirm that too, but the thing with glycogen is it is a branched structure. It is heavily branched, and the properties of it is due to the extensive branching in its structure, not because of hydrogen bonds present in it. So I think the answer is 1, 3, and 4 here. Though hydrogen bonds can be present inside the structure of glycogen, but it does not define its properties. Whereas if hydrogen bonds are absent in hemoglobin, the structure will be distorted and the properties will be lost. Moving on to another question. Some features of triglyceride are listed. So, triglycerides can be liquid or solid at room temperature. This is true. For example, butter and oils. So, saturated triglycerides are solid at room temperature and unsaturated ones are liquid at room temperature. So, this is correct. Contains high proportion of carbon-hydrogen bonds. So this is also correct. Insoluble in water. This is also correct, and less dense than water. So this is also correct, as you have seen that oils, for example, if you drop them in water, they makes their a layer on the surface, which means that they are less dense than water. So, which of the features makes triglycerides suitable for energy storage? So, okay, all of them are correct, but which are the best choices for making them suitable for energy storage? So, they contain high proportion of carbon and hydrogen bond. This is the first thing that you will see as being relevant in this question. So 2 is basically should be in the options. So here we have in A we have 2, B, C we have 2, and D we have 2. So B is incorrect. Now, insoluble in water. Does this accounts for anything which is you can say relevant in this question? I don't think so. Solubility in water doesn't account for it being a suitable for high energy compound, or maybe let's just see less dense than water. Okay, so the fourth one is definitely you can say fourth is definitely irrelevant. Can be liquid or solid at room temperature. This is also irrelevant for me, I think, because either it being solid or either it being liquid doesn't accounts for it being a suitable source of energy, because if it goes inside the body, it would be broken down into small pieces, yeah, so it would not be in the form of, let's say, polymer, yeah. So how can I explain this? So let me just change the approach here. Let's just start this again. Can be liquid or solid at room temperature. So if it's solid or either if it's liquid, this does not account for it being a suitable energy storage, or you can say a suitable energy resource. So I think this one is incorrect and this is irrelevant. Contains high proportion of carbon and hydrogen bonds. This is definitely a plus point, or you can say one of the advantages that you can say triglycerides have over the glucose. That's why they release high amounts of energy, which is double the amount of energy. And soluble in water, I'm going to explain it in a bit, but this point, less dense than water, this is irrelevant here. Now 2 and 3 can be the answers, and I think 2 and 3 are the answers. Now you're going to ask, why does insolubility gives it a feature or a property of being a suitable energy resource? Now I'm going to explain you why. For example, this is blood vessel here, and it contains different kind of red blood cells and plasma, etc. Now, what happens here is that there are some molecule of glucose. Now, as you guys know, glucose is a soluble substance. So if the concentration of glucose increases to an extent, it's gonna make the osmotic potential of blood very low, or you can say water potential of the blood will become lower, or the blood will be more saturated, thick, sticky, and this is a very dangerous thing. So to cope up with that, blood sends insulin, which controls the, you can say, glucose concentration inside blood and prevents it from being, you can say, accumulated, or you can say thickening the plasma of blood, because it is soluble. And you can take it for an example, like there is a glass of water in a cup and you keep adding sugar again and again and again. There will be a point that there will be hypertonic solution, which means that the solution will be very concentrated, and this happens the same with the case of water or blood. But if you consume triglycerides, for example, and they directly goes into your bloodstream, so if there are molecules of triglyceride, because of their insolubility and their insoluble structure, or you can say they are being non-polar and they don't dissolve in water, this property of theirs will not affect the osmotic potential of blood. So the water potential inside blood, or the osmotic potential of blood, remains same. So this is one of the advantages, that triglycerides doesn't increase or decrease water potential, and because of that they are a suitable source of energy storage. Anyways, coming to question number 12, which is some food contains hydrogenated vegetable oils. These are unsaturated, means double bonds, fats that have been converted to saturated fats to single. Which property of fats will have been changed? So as I've told you before, double bonds makes liquid and single bond makes solid. So the hydrocarbon chains will fit more closely. This is true. Their solubility in water will increase. They are insoluble in water regardless of double bonds or single bonds. They will have more double bonds than in their molecule. This is absolutely wrong, because saturated fats have no double bonds here. They will remain liquid at room temperature. This is also wrong, because they become solid once they are saturated. So the best possible answer in this condition is A, or the correct answer is A. Question number 13. Which statement could be used to describe enzyme molecules and antibody molecules? So, enzyme molecule is a protein, and antibody molecule is also a protein here. So, hydrogen bonds stabilize, and both are tertiary structure. Hydrogen bonds stabilize the structure of protein and are important to its function, for it to function efficiently. So this one is correct. Hydrophilic R groups point to into the center and molecule of the molecule and cause it to curl into spherical shapes. So this is correct too. The tertiary structure of the protein molecule plays an important role in the functioning of protein. The tertiary structure of protein molecule plays an important role in the functioning of protein. This is also correct, because let's say in the case of enzyme, when it denatures, the hydrogen bonds between it breaks, and because of the breaking of bonds, it loses its shape and its role. So all of those statements are correct for me. And we'll check those answers once we have done 20 questions. We'll go and check that if we have done anything wrong. A student carried out 4 tests for biological molecules on a solution. The results are shown in the table. Iodine test. Again, this question refers to the tests here. So it's not really important to do it. Or should we do it? I don't know, but anyways, let's just say in burette, due to its purple color, protein is present here. Orange color and clear, so there are no fats. So okay, this is correct, and sucrose. So should I attempt this question, or maybe I should just leave it for the part? Okay, let's just write it here. Question number 7 and 14 here are of the tests. A student, okay, again Benedict tests comes, so 15 also refers to the testings. Which raw shows chemical groups present in biological molecules listed? So biological molecule is amino acid, presence of carboxyl, C11OH, yes. So in amino acid, NH2CHR group C11OH, which is the carboxylic group, is present inside the amino acid. So yes, and the presence of 2 or more hydroxyl groups, no. So this is the raw one is correct in everything. There is no OH present. There is not more than 2 OH groups. There are only C11O and one OH group. So hydroxyl group, only one hydroxyl group is present. As we talk about beta glucose, it does not contain carboxylic group. So this is correct. And it is having 2 or more hydroxyl groups. So yeah, so second raw is correct too. If we talk about the third one, glycerol does not contain C11OH, and it does not contain 2 or more hydroxyl groups. So let's just see here, C, and C. So this was the structure of glycerol here, and we have 3 hydroxyl groups. So this raw is incorrect. Fatty acids, we have C11O obviously, because it contains the you can say acidic group, which is carboxylic group, and it does not contain the hydroxyl group, 1 or 2 or more hydroxyl groups. So this is correct too. So 1, 2, and 4 are the answers here. Or if we think about proceeding with the elimination method, so because 3 is incorrect, so A contains 3, C contains 3, and D contains 3, so the only thing that we are left with is B. If you were a bit confused about fatty acids, then you can do with this method. So question number 17 says, so this is glycerol, these are fatty acids, and this is triglyceride. Okay, so which type of bonds are formed in macromolecule product? So these are ester bonds, because the bonds between alcohol and carboxylic acid are ester bonds. It's not glycosidic, this is not hydrogen, this is not peptide bond here. So A is the answer. Fibrous protein are composed of chain of amino acid held together. An example of fibrous protein is shown. Which type of bonds is at X and Y? So Y contains a molecular bond within the molecule, intramolecular bond, which is the molecule which is formed between two atoms, and X is intermolecular, or you can say the bonding between the weak interactions between the molecules, which is, in this case, this is hydrogen bond. Sorry for my handwriting here. And this is peptide bond. So Y is peptide and X is hydrogen. So B is correct. Coming to question number 19. Oh my god, let me just see how many questions are there. So there are 166 questions. We're gonna do a marathon after we have done enough questions. Let me just see if we will do till 50, and then we will just go into marathon mode where I'll be just reading and you'll just are just marking, and will not be explaining much because there is gonna be repetition. Anyways, going to 19th question. Which row about the structure of protein is correct? So primary structure, there is the number of amino acid present in protein, is the order of amino acid present in protein encoded by DNA, is the result of translation of mRNA molecule by a ribosome into a chain of amino acid, is the sequence of amino acid in a protein coded by mRNA molecule. All of them are partially correct. Is secondary, is the left-handed spiral formation of primary structure, is the coiling of chain of amino acid to form beta-plated and alpha helix sheets. This is correct. Occurs because of the attraction between hydrogen and oxygen in the peptide bonds. Wait, this is not correct, I think. Is formed by hydrogen bonding between amino acids, forming the primary structure. So this is correct too. Let's just talk about quaternary structure, which is the interaction or bonding between two different kinds of polypeptides. So is the subunit of polypeptide that links together to form a protein. So this is correct. Contain two types of polypeptides that interact, forming the shape of protein. Okay, so this is also correct here. Is formed by 4 polypeptides and an additional reactive group attached to the protein. So this is talking about hemoglobin, which is correct for the quaternary structure, but this is specific, yeah? So every quaternary structure does not contain 4 polypeptides. So I think this is wrong. Is formed by linking together one or more than one polypeptide to form a protein. So this is correct too. Now we are left with B and D here. So let's see what B completely says and D completely says. So in B, it says is the order of amino acids present in a protein encoded by DNA. So this is wrong here. You know why? Because, and this can be partially true because, I mean, technically if we talk, the coding is done by the DNA, but the mRNA molecule provides the codes for the ribosome to code in a sequence. So I think the D is more correct here, or you can say best choice here, because it accounts for the sequence of amino acids in a protein coded by mRNA molecule is formed by hydrogen bonds between amino acid forming the primary structure. and is formed by linking together one or more than one polypeptide to form protein. Now, B could also be true, and we will see maybe it is possible that B is correct here, but for me, the best possible answer is D. Going towards question number 20, a student wrote 4 statements about water. Water has high specific heat capacity, which maintains the temperature of water within cells. Mammals rely on water having a relatively low latent heat of vaporization to keep them cool. When a negatively charged ion is added to water, as positive charge on hydrogen ion is attracted towards the ion. Water, when surrounded by water, non-polar molecules tends to be pushed apart from one another. Which statements are correct? So, water having high specific heat capacity to maintain the temperature of water within cells. This is correct. So number 1 is correct. So this is wrong. This is wrong. Mammals rely on water having a relatively low latent heat of vaporization. So this is wrong, and it is wrong in both of the statements because they have high heat of vaporization, which helps for the cooling effect, as I've explained in the chapter of biological molecules, where I explained the properties of water. The third one says, okay, we don't really need to read the third one because it's correct in both, but what makes difference is going to be the fourth. So water is surrounded by so when the water is surrounded by non-polar molecules, which are hydrophobic molecules, the non-polar tends to be pushed apart from one another. So pulling means attraction and pushing means repulsion. So it is correct. So the best possible answer here is A. And let's just check the questions here. Let me just pull up. So yeah, let's just see. So question 1 is D, 2 is D, 3 is D, and 4 is A. So let's just mark those questions. So 1 is correct, 2 is correct, 3 is correct, 4 is correct. The 5th one says CBBDAC. CBBDAC. So 5th one is correct. 6th one is correct. 7th one we have left because I will explain it in the next class. So 8th one is correct. I think 8 is D and 9 is E, A, and 10 is C. So 9 is also correct and 10 is also correct here. Going towards the 11th, so D, A, C, and 3 B's. So D is correct, A, and okay, so 13 is wrong here. And 16th is correct. A, B, D, okay, so let me just close this. So yeah, So one of the answer was wrong here, which is this one that I need to correct. Let me just mark it with red. So the question says, which statement could be used to describe enzyme molecules and antibody molecules? So the answer is, the correct answer here is C here. So this one is the correct answer. This is 1 and 3. So let's just read. Hydrogen, so 1 is correct, we know, but why is 2 incorrect? Let's just, hydrophilic R groups points towards the, oh my god. Oh my god. This, this is the thing. And it have happened with me a lot of times in the past too. I tried to be attentive while doing the question, and even though when I was doing this question, I repeated the statement that hydrophobic points towards the center and hydrophilic points towards the surface in a you can say water-based solution. When the, for example, you can say phospholipid is you can say dropped into water, or you can say reacts or interacts with water. The side which is hydrophobic tends to move towards the inner side or center, and the side which is hydrophilic goes towards the surface. And I repeated this twice. Still, I read this hydrophobic and considered it to be right. Anyways, the rest questions are correct. And this is the thing, guys. I mean, you need to be very attentive while solving the questions, because sometimes your brain makes you think that this is hydrophobic written here, and you thinks that it is correct, but this hydrophilic word spawns randomly and you are like, yo, it was not before. Anyways, the 21st question is basically based on the tests. What occurs during the formation of glycosidic bonds between two alpha glucose molecules? So alpha 1,4 bond is formed. Say a hydrogen bond is always formed. Not necessarily. A molecule of water is always formed. This is also correct. A hydroxyl OH group is always formed. This is incorrect. No, I need to read this again. So, one thing you need to think about is, if we talk about a specific molecule here, which is, let's say, glucose. So for example, this is glucose here, and it forms a glycosidic bond with another glucose, alpha glucose. For example, this is the worst drawing that I've ever made, but this is 1,4 glycosidic bond which is formed, but in the glycogen and amylopectin there is 1,6 glycosidic bonds present too, and in the glucose and fructose glycosidic bond there is 1 and 2 carbon involved in the formation of glycosidic. So 1,4 is not always formed. even, I mean, the fructose example was irrelevant here because here's clearly written that two alpha glucose molecules form glycosidic bonds. But what about 1,6? 1,6 glycosidic bonds also form between two alpha glucose. So 1,4 is not always formed, but one thing which is always formed is the water molecule. So what is the maximum number of condensation reactions that occur in a triglyceride in its synthesis? So number of condensation reactions, so there are going to be 3, because 3 fatty acid chain reacts with 3 you can say OH molecules to form 3 water molecules. So you need to remember, I have done this question wrong when I was doing my A levels. So there was a question where it was written how many water molecules are formed, and because I was in a hurry and I saw that condensation reaction occurred between triglyceride, you can say occurred between fatty acids and glycerol, forming a one molecule which is triglyceride. I in a hurry I just said that one water molecule is released, but there were 3 water molecules that were released. They need to remember that. And let's just take a break, a short break, 5-minute break, and I'll be just coming in a minute. So let's just start again. So 3 proteins have quaternary structure that have quaternary structure are listed. Type 9 collagen is formed from 3 different polymers. The main form of hemoglobin contains 2 alpha globin and 2 beta globins. HIV protease consists of 2 identical polymers. So, three proteins that have a quaternary structure are listed. Okay, type 9 collagen is formed from 3 different polymers, so 3 chains are involved. The main form of hemoglobin contains 2 alpha, so 4 chains. And HIV protease consists of 2 identical polymers. So the protease enzyme inside the HIV, which is a virus, contains 2 identical chains. Which row shows the correct number of genes needed to code for each of protein? So for hemoglobin, 2 genes are required. For HIV protease, 1 gene is required, and for the type 9 collagen, 3 genes are required. So it's going to be 3 to 1. Type 9, 3, 2, and HIV 1. And what is the reason for that? Let me just explain. It's very easy, by the way. So 1 gene here accounts for the production of 1 chain of amino acid. Now, type 9 collagen is formed from 3 different polymers. Yeah, so that means here 3 genes will make 3 different chains or 3 different polymer of protein. In hemoglobin, though there are 4, but there are 2 alpha and 2 beta. So that means alpha is made from 1 gene, even though there are 2 strands being made for the protein, but there is going to be 1 gene used for alpha and 1 gene is going to be used for beta. And for the HIV protease, 2 identical polymers, which means that 1 gene is only required for the formation. 1 gene only has the code for it, so 1 gene will only be used for making 2 different kinds of protein. Though I have not explained translation and transcription at this point, because it was not really necessary in this chapter, but you will be able to do questions like that easily and after that chapter, because listen, this, even though these are topicals, yeah, but these are from the paper of Cambridge, yeah, which considers while making exam that you have done the complete syllabus. So even if some highlights of another chapter are gonna be in it, but the concept, the core concept, is gonna be the same. So the diagram shows the structure of amino acid with some parts of molecules that are shaded. Which diagram shows the shaded area of the molecule that is common to all amino acid? So the common part in all amino acids are NH2, CH, and C double bond O and OH. So this is the common part here. CH3 is not common. CH3 is not common at all. CH3 is not common. Okay, wait, hold on. Which diagram shows the shaded part of molecule that is common to all amino acid? So CH3 is shaded here, so it is not common. It is shaded here, it is not common. Here it is not shaded, so we need to see CH, C double bond OH. NH2 is also common, so we need to see, and we can definitely see that the only structure that resembles with this structure of ours shaded is this one, B. Now, going to 26. And after 50 questions, after we're done with 50, I'm gonna take a break, a half-hour break maybe, and we're gonna solve those afterwards, or maybe tomorrow. Let's just see how much potential do I have. So this is regarding the practicals. A student carries Benedict test. Again, this is a practical question. What can occur during condensation of carbohydrates? Okay, one thing I need to tell you. This is important in A levels, yeah? So the tests, all the Benedict and Buret, I'll be doing it. I'll be just explaining that in the next class maybe, but before CIs I will just explain that. But for this part, which is this experimental, this is not required in IMAT, or you can say MCAT, in any case, yeah? So these are not required. I've seen the syllabus. Anyways, what occurs during condensation of carbohydrates? So carbohydrates, polysaccharides are formed, disaccharides is produced from monosaccharides. This is correct. Glycosidic bonds are broken. No. In condensation, the glycosidic bonds are formed. Molecules of water are used up. No. Molecules of water are released in condensation. Monosaccharides are produced. So this is also correct. This is also wrong, because this is the hydrolysis, what we are talking about, in which monosaccharides are produced. So going to the next question, the diagram shows 3 hexose sugar. Now this is the question where I told you to memorize these structures. So let's see here. Obviously the third one is known to everyone because it's very easy. The third one is so which row correctly shows example of carbohydrates in which these 3 hexose occurs? So in sucrose, so let's just name this. So this is fructose. This is carbon 1, 2, 3, 4, 5. So up, down, up, down, up. So this is alpha glucose. Down, down, up, down, up. And down, down, up, down, up. So this is gonna be the beta glucose. So in cellulose, number 1 is gonna be involved. But wait, there is no number 1, so I am wrong at this point. Beta glucose is this one. I was also confused in it. Beta glucose, and this is alpha glucose. So repeat after me. Alpha glucose is down, down, up, down, up. This is alpha glucose, whereas beta glucose is up, down, up. And we can see fructose here. So cellulose is 2, 2 here. And it is not 3, whereas in starch, it is alpha glucose used. So the number 1 is used. And 1 here is used, and 2 is not used, and 3 is not used. Whereas in sucrose, 3 is involved, 2 is not involved, 1 is not involved. So only option with all 3 correct options are D, is D. Now, what is true about triglyceride? So triglyceride are hydrophobic. Are triglyceride hydrophobic? Yes, they are. They are insoluble in water. If they were hydrophilic, they were soluble in water. So yes, this is correct. Now, this is the question that when I was giving AS, or when I was in AS, I did this question. It usually came in our mock examination, which is school-based. It's not related to CAIs. So It came in first term, I did it wrong. It came in second term, I did it wrong. And it came in the mocks, which are before CAIEs, a practice examination, and I did it wrong again. I mean, that question came 2 times in the last paper, and I was confused in that, and I did, for example, A in first question, and when it was repeated, I did another option because I was uncertain about it. But anyways, it is insoluble in alcohol. So, alcohol is polar, yeah? And alcohol is the solvent for alcohol is non-polar and is a solvent for non-polar molecules. So the fats, or you can say triglyceride, are gonna be soluble in the alcohol, but because it's written insoluble, then it is incorrect. So this B is the correct option here. What is the maximum number of carbon atoms in an amino acid? So how many carbon atoms are there in an amino acid? So NH2, CH2, or you can say CH or R group, and C double bond O and OH. So 2 carbons are there. So this is easy. After boiling a sample of milk, Benedict solution of yellow color. It's not present, by the way, but the question is not gonna be solved by me. So which of the 3 statements about polysaccharide can be used to describe amylose and cellulose? So amylose is the straight chain of alpha glucose, whereas cellulose is straight chain of beta glucose. It contains high amounts of hydrogen bonds, where it does not necessarily contain so much amounts of hydrogen bonds, but it is not asked in the question. So what's asked in the question is it contains 1,4-glycosidic bonds. Which statement about polysaccharides that can be used to describe amylose and cellulose? So both contain alpha 1,4. So both contain 1,4-glycosidic bonds. None of them contain 1,6-glycosidic bonds. And polymer of glucose. So they both are polymer of glucose, but the only difference is cellulose is made up of beta glucose and amylose is made up of alpha glucose. So it's not mention of alpha and beta here, so 1 and 3 are going to be correct options. Which diagram shows the formation of peptide bond, NH and OH? So this shows the peptide bond. This doesn't show a peptide bond, this doesn't show peptide bond, and this doesn't show peptide bond here. So the only correct option here is A. I don't need to repeat how peptide bonds form because we have done this question in the beginning. So which feature affects the tensile strength of collagen? So the helical structure of collagen chains, the small R groups of amino acid and collagen, the insoluble nature of collagen, the bonds between collagen molecules. So this is a quick question here. So which thing is gonna affect the strength of collagen? Which is this? It is a secondary structure, yeah, because it is a structural protein, so they are mostly secondary in their structure. So helical is the secondary structure, the helical structure of protein, the collagen chains. This is going to affect the tensile strength. The small R groups of amino acids in collagen, this is also going to affect, obviously, because hydrogen bonds are formed very extensively. The insoluble nature of collagen, let's just consider that. The bonds between collagen molecules. So the hydrogen bonds that forms between collagen molecules are important. The insoluble nature is important for tensile strength. Let me think about that. So if it was soluble, will the tensile strength be affected? Let's just see. Let's just use elimination method. So 1, 2, and 4 are correct here. So 1 is not here, 2 is not here, 1, 2, 4, 1, 2, 3, 4. So I think that it is unnecessary to consider insoluble or solubility of collagen, but I would do it otherwise. Answer should be A, because if it was soluble and water pressure or hydrostatic pressure was on the cellulose molecules or collagen molecules, it would have been dissolved and lost its tensile strength. So it can be a point, but let's see what does the marking scheme says. It is again the question of experiments. Let's go. The diagram shows 3 hexose sugar. This is repeated. And as you can see, sucrose is third, 2 and 1. So this is again repeated. You need to remember both of those structures. You need to remember the structure of galactose too, because these 3 are very confusing: alpha, beta, and galactose. What can occur as a result of condensation reaction? Disaccharide is formed, glycosidic bonds are broken. Now, so again, this is again repetition. A water molecule, a molecule of water is produced. 2 monosaccharide joins together. So 1, 2, and 4 here are correct answers. Is broken. So 2 is correct. 2 is incorrect. 2 is incorrect. And 2 is incorrect. So 1, 3, and 4 are the answers here, because glycosidic bonds forms during the condensation reaction. So the diagram shows some relationship between features of carbohydrates. So alpha glucose, 1,4 bonds and branched. Okay, so which raw correctly matches for carbohydrate with some of its feature? So alpha glucose and 1,4 glycosidic bonds. So it's definitely gonna be amylose. If alpha glucose 1,4 and is branched, it's gonna be amylopectin, or it can be glycogen. So alpha glucose and 1,4 bonds are usually, so the number 1 is amylose. It can't be cellulose because it's mentioned here, it's alpha glucose, so it can't be collagen either, and can't be amylopectin here. So because amylopectin and glycogen, both of them are you can say branched structure, and one doesn't involve branching, whereas if there was starch, then we might have considered, or we might have thought of it being added, because you can say starch does contain amylose. So yeah, so B is the correct answer in this question. Which raw describes a triglyceride? So triglyceride are hydrophobic, so these 2 are correct, and they are soluble in alcohol, so both are wrong here. I think this question came and I did it wrong twice. Which feature of collagen results in it having tensile strength? Bond formation between adjacent molecules, yes. Each 3-strand molecule is held together by hydrogen bonds, yes. Every third amino, what's amino acid doing in the collagen? Okay, so amino acid, yes. So every third amino acid is in polypeptide is small. So this is not mentioned. I don't know if it's correct or wrong. The primary structure is held together by peptide bonds. So the high tensile strength of collagen is not because of the peptide bonds between the between the amino acids in the you can say primary structure, because it's present in every protein. So we're not going to say that every protein is as you can say strong as the collagen fiber, because all of them contain peptide bonds. So this is going to be wrong. So 1 and 2 makes sense. 4th is wrong, so 4th is wrong here, 4th is wrong here. Now we need to think about the 3rd one. Mm, I think that we should leave it, because if or maybe I would just do C here, and let's just say, is it C? So 41 is A. So this is the correct answer here. So this is the fact that you need to remember. Every third amino acid in polypeptide is small. So in collagen, every third amino acid, although one thing I need to tell you, that there are repeating sequences that I've told in the biological molecule chapter explanation, that the sequence are repeating units. But there's one more point that you learned today, that every third amino acid is polypeptide of the polypeptide in collagen fiber is small, which accounts for the high tensile strength. So let's go towards another question, which is, the diagram shows 3 glycerides, X, Y, and Z. So these are 3 double bonds, 1 double bond, and there's no double bonds, of saturated and unsaturated. These both are unsaturated. This is unsaturated, saturated, and unsaturated. These all are saturated. So what is correct for these 3 triglyceride? Contains saturated fatty acids. So all of them contains saturated fatty acids. So XYZ contains unsaturated fatty acids, contains unsaturated fatty acids. So X and Y contains unsaturated fatty acids. So technically A is the answer, but we are gonna read the last part. Contains more than 2 different fatty acids. So both of them, X and Y, contain 2 different fatty acids which are saturated and unsaturated. So this is correct for this 2. So the answer is going to be A. 43 is, which features of glycogen distinguishes glycogen from starch? So glycogen is extensively branched, more branched than the amylopectin in the starch. So all glycogen molecules are highly branched. This is correct. All glycogen molecules are polysaccharides. So this is not the distinguishing feature. All glycogen molecules contain alpha glucose. This is also not the distinguishing feature, because starch is also made from, or is the polymer of alpha glucose. All glycogen molecules contain 1,4-glycosidic bonds. So this is also not the distinguishing feature, because amylose and amylopectin in starch also contains 1,4-glycosidic bonds, but extensive branching is in glycogen. So 4 solutions were tested with Benedict. So this is the question of practicals. Going for the diagram shows the molecule of 3-hexose sugar. Again, this is repeated. I think, no, okay, this is not repeated. So this, the 3 is the third one is the sucrose. So sucrose here, wrong. Cellulose is the second one, because it is beta glucose, up, down, up. And amylopectin, because it's made from alpha glucose, it's this structure. And let me just confirm this, because maybe if I'm wrong, maybe for hold on, let me just So yeah, let's continue. Or maybe we should check answers after the 51. A student wrote 4 statements about what happens during hydrolysis of a carbohydrate. So hydrolysis is breakdown. So disaccharides are formed from monosaccharides. This is wrong. Glycosidic bonds are broken. Yes. My molecules of water are released. No. And monosaccharides can be formed. So yes. So 2, 2, 4, and 2 and 4 are correct here. 47 says the table compares 3 molecules XYZ which contains the element carbon, hydrogen, and oxygen only. The percentage of carbon, hydrogen, and oxygen in each atom, oxygen's atom in molecule is shown. So X is in X 25% carbon, 50% hydrogen, and 25%. This is definitely glucose. 28.5% carbon, 47.7% hydrogen, and 23% oxygen. And here carbon and hydrogen are high and oxygen is very low, so this is going to be fats or triglyceride, you can say. So X and Y we know, X and Z we know, Y we're going to know in a bit. So X is monosaccharide, polysaccharide, it's not a polysaccharide because the composition is exactly CH2O. But in the polysaccharide, because of the extraction of H2O from the molecule of glucose, so the percentage is not exactly 50, 25, 25. So it's monosaccharide. Y is polysaccharide, or you can say disaccharide, whatever you want to call. Both are correct here. And the last one is not, Z is not polysaccharide, but it is triglyceride because the percentage of oxygen in it is very low. So because of that, it is a fat molecule. Anyways, which statement about the difference between phospholipid and triglyceride are correct? So one thing I must mention here is that disaccharide have a bit higher percentage of hydrogen and oxygen than that, because in disaccharide, one OH from the left and H from the right side, OH from the right side, they both are present, and only one OH group is absent, and then there's only one H2O molecule extracted. But in polysaccharide, due to many hydrogen and H2O molecules being eliminated, each glucose molecule is deficient of 2 hydroxyl groups. So for instance, this is a glucose molecule, and there are glycosidic bonds at 1 and 4th carbon. So OH molecule is going to be eliminated from here. OH molecule is going to be eliminated from here in order for the formation of the glycosidic bond. Whereas in the disaccharide, only OH molecule from one place is going to be displaced, making a percentage a bit higher than that. Anyways, moving on, the triglyceride, as I've explained, there is very low composition of oxygen, and this is what is the difference. The high percentage of CH bonds and very low percentage of oxygen present inside the fatty acids, or you can say in the triglyceride or fats. Which statement about differences between phospholipid and triglyceride are correct? Phospholipid have a hydrophobic region and triglyceride do not. This is correct. The fatty acid in phospholipid. In a phospholipid are always saturated, but in triglyceride they may be saturated or unsaturated. This is incorrect. And phospholipid are polar molecules, but triglycerides are non-polar. So phospholipid head on the phospholipid, or you can say the phosphate head on phospholipid is polar. But if we consider the molecule is both polar and non-polar, the polar head and the non-polar tail. But because it is both polar and non-polar. The whole molecule is not polar, so I don't think so it should be considered a valid point. So the fatty acid and phospholipid are always saturated. I think this is not correct, so one is correct only. I think maybe I'm wrong here. If there was option 1, 3, I should have considered it, but an investigation with blah, 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 it is a experimental test again. Which level of protein structure can we determine by determine the specificity of enzyme? So the protein structure that determines the specificity of enzyme is the tertiary structure. Quaternary structure, let me think, can be secondary. So no protein is secondary structure, and primary is obviously not. So what should I do? I don't think there is any enzyme that is quaternary in structure, so I think the answer is going to be 1, 2, and 3. Now let's check this. Maybe I'm wrong. $50 is A. 1, 2, 3, 4. So okay, all are correct. So quaternary structure. So there are enzymes that are quaternary in structure. So yeah, all of the levels are important. So let's just check from 20 here. So B, 3C, and B. C, 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 and B. 3A and DB. So 3A and DB. 3 B's, A, B, and 2. So B, it is not unintended, and B for 34 is A. 35 is okay. So this is wrong here. I need to mark this as wrong. Wait, 35 is B. Okay. 36 is not attempted. 37 is D. C. 39 is B, and 40 is D. Okay, 40 is D. 41 is A. Hurrah, I did this correct. 41 is A. 42 is A. 43 is A. 44 is again unattempted. 45 is D. 46 is C. 47 is B. 48 is D. 48 is D. Okay, so this is incorrect. 49 is B and A. Okay, so B and A. So what's incorrect in this? So 48 is D here. So 3 is only correct here. Oh my god, I again messed up this. So in A it's written that it contains hydrophobic region, and triglyceride does not, whereas triglyceride is whole hydrophobic, whereas phospholipid have hydrophilic region. Oh man, I didn't read this hydrophobic. I thought it was gonna be hydrophilic. Anyways, so again, my misunderstanding in this and what's in that. What feature affects the tensile strength of collagen? So 35 is B. So yeah, I left this unchecked because I was unsure about this too. So the third one, the insoluble nature of collagen is not necessary for the tensile strength of collagen, though I thought it could be, because if you want to because the tensile strength of collagen is more usable, if because a lot of parts in our body are based on water, yeah, and if there is a molecule which is water soluble, which is collagen, then it's no use, obviously, because the strength is of no use because the molecules are going to be dissolved. But it might be correct here because it not usually accounts for the tensile strength. The tensile strength is due to the structure, and I was also considering that, but I thought I changed my mind at the end, and I said that I'm unsure about this question. This was a good question, anyways. So we have done 50 questions till now, and our stream is 1 hour and 50 minutes. So I think we should spend 2 hours more and complete this chapter. We should start marathon. What do you think? So let's just take 5 to 10 minutes of break, and let's just continue after that. So quick update. Let's just end this today and continue this tomorrow. I think 60 questions are left hardly, and we'll be just covering those in today. It took a lot of time, I think 1.5 hours. Tomorrow it will take 30 to 40 minutes, because I'll be just solving the questions quickly and explaining the ones that are required to be explained. And then we're going to start the cells and do maybe around 30-40 questions of cells tomorrow. So until then, bye bye.